All right, so hi everyone. I would like to show my respect and acknowledge the traditional custodians of the land, of elders past and present, which this meeting takes place. It's my great pleasure to introduce the Associate Professor Susana Herculano Russell. So Susana is a Brazilian-born neuroscientist, currently working at the Vanderbilt University in the United States. And one of her main interests is to understand the brain diversity, as you can beautifully see here. And particularly at the interface between metabolism, longevity, and cognition. And in addition to her wonderful work in science over the last decades, her ability to communicate science is very impressive. And if I can speak in name of of the young generation of Brazilian researchers, I can surely say that Susana is a big reference for many of us in my country. So when I knew that she'd be here in Australia, I did not hesitate to invite her to come here in Melbourne and give a talk to us. And thanks very much for accepting our invitation. And now it's over to you. Thank you.
Thank you so much for Pedro for the kind introduction and the prompt invitation upon my announcement on Twitter. Hey guys, I'm going to be in Australia. Anybody wants me there? So Science Twitter is a wonderful thing, and it's thanks to Science Twitter and Pedro that I'm here today, which I'm really happy to do because I love every single opportunity I have to talk about my work and tell my stories and run them by people who have zero obligation to know the first thing about what I'm talking about, because I'm really interested in your questions and your comments and your opinions, especially because what I'm going to tell you today is actually, yeah, you're the first audience ever to get this story. So, and the story I'm telling you is I came to realize we've been hearing for the longest time that caloric restriction and exercise are the two tried and true ways to live longer. And most importantly, to live healthily longer, right? And yes, it works in C. elegans, it works in fruit flies, but it also works in vertebrate organisms that have a brain with a hippocampus. And many of the effects related to caloric restriction and exercise that prolong healthy lifespan, they have to do with improving the health of hippocampal neurons through increasing the production of BDNF. But interestingly enough, people don't question why, which I didn't either. I only came to realize that, wait a minute, what sense does this make that manipulations that increase BDNF in the hippocampus would lead to a longer life? And yes, of course, maybe the increasing BDNF in the hippocampus is the one thing that you're seeing, and there's a number of other effects that you're not seeing. But the point remains that you always see hippocampus, longevity, caloric restriction and exercise and BDNF together in the same paragraphs and the same papers. But what does one thing have to do with the other? What could the hippocampus in the brain possibly have to do with longevity? So I could tell you a story of how I've realized that all of that actually makes great sense, but that's not how things happen. And the older people in the audience know that damn well at this point, but the younger people don't. So I want to make sure that I give you historically accurate account of how do you discover that something makes sense when you didn't have the first clue about that to begin with. So let me start with, Pedro mentioned that my main interest is diversity, and that's exactly right. More specifically, I'm really interested in the diversity of brains and nervous systems as a whole. And that expands very rapidly to life as a whole once you realize that you don't need a brain to be alive, but that's a whole other story. My interest is centered around three main questions. What is the extent of diversity? So how far can diversity go? What are the limits to diversity? Are there places where brains just do not go? And does that mean that they do not go there because they cannot? So that are there rules that determine what is the range of possible? And third, and most important for today, so what that you have diversity? So what that some brains are much bigger than others, have more neurons than others? And especially, let me jump to one part of the brain, which is what most vertebrate neuroscientists tend to have in mind when we say brain, and that's really the cerebral cortex. So why do we bother with the cerebral cortex so much? And the hippocampus is a part of the cerebral cortex. Why do we bother so much that I'm going to tell you about a method that I developed to be able to count how many neurons cortices are made of in different species, and what difference does that make? So we care. This is my take on it. When I teach neuroanatomy, this is what I tell my students. So you're getting the 2-minute version of my whole neuroanatomy course. We care about the cortex because we actually don't need it. And what I mean by don't need it is that the parts of the brain that really operate the body, that really keep us alive at any moment in time, you find them here. You find them in the brain stem, the axis of the brain, all the way up to the very front of the brain, which is the hypothalamus, not the cerebral cortex, a whole other story there too. But these are the parts of the brain that make things happen in your body and represent back what happens in your body. Now, you notice the cortex doesn't do any of that. But the cortex, which we traditionally associate with cognition and higher functions, it does do cognition and a bunch of interesting higher functions, not because it talks directly to the body, or maybe exactly because it doesn't talk directly to the body. What the cortex does is it receives copies of everything that's happening down here, relayed to it by this structure, the thalamus. But we're not done yet. The most important thing is that the intrinsic circuitry of the cortex is highly associative, meaning that the cortex has the possibility of bringing all these signals from different parts of the brain together, and it's capable of forming associations that weren't there before. And the hippocampus in particular is smaller part of the, well, it's a part of the cortex that sits right here. It develops from the same part of the neural tube that forms the cortex as a whole. But the key is the hippocampus is this lower part in the posterior cortex that is strongly connected to a lot of these structures here that do operate your body and allow your body to function as a whole. And more than that, integrated as a whole, that is integrated with your mental reality, with your current experiences, with your recent past experiences, with your projections for the future. So I'd like to talk about the hippocampus, not as part of the limbic system. That's another whole other story there. There's really doesn't exist. Oh, okay, back to where I was. I'd rather think of the hippocampus as the key structure of an integrative system in the brain. So what's so important about this integrative function that cortex as a whole and hippocampus in particular have within the brain? What do animals that have a cortex and a hippocampus in particular have that other animals that in other forms of life don't have? And I'd say it boils down to this one word, which is flexibility. Flexibility that emerges from this capability of associating events, of bringing things together and creating new things in the process. For example, with flexibility comes complexity, different ways of doing different things. With complexity, one of the forms of shapes that complexity assumes is the adaptation, the ability to adapt your behavior, to change your behavior according to what has happened in the past, which has been used as a definition of intelligence, but that leaves out an enormous achievement of flexibility, which is the capability of acting ahead, of acting now towards some simulated future state that hasn't happened yet, but that you can emulate and present to yourself as a possible reality that can then guide your behavior. That means that in the process, you gain a past that you can represent. And because you have a past, you have now the possibility of simulating futures, of simulating, projecting the different versions of that past into the future, which can guide your actions. So all of that makes your behavior flexible. So flexible that you can simulate not only your own past and possible futures, but others as well. And that is when we gain the possibility of internalizing other people's minds, of representing what's going to happen to others when we organize our behavior, which is the basis of life in society, which is in turn the basis for our capability to form a joint culture that transcends the individual and that can be carried on to the next generations. So put all of this together, and the one of the basic premises of everything that I'm going to tell you next, of course, is that the more neurons you have in the circuits that do all this, the more of all this you should be capable of doing, right? Which brings us, of course, to cognition, which is the first thing that people think of when they think cerebral cortex or hippocampus in particular. So you would expect that the more neurons that a cortex has, that a species has in the cortex, the more that species should be capable of doing. And neuroscience as a whole is slowly starting to collect that kind of data that allows these questions to be addressed. And what I'm showing you here is, I won't go into the details, but you're essentially looking at performance in a task that requires self-control, that different investigators in lots of different countries around the world posed to their own species of expertise. And they all asked these species to, they tested if the species were capable of withholding a behavior, which was reaching straight ahead for a piece of food. And once the food was presented inside a transparent cylinder, how long did it take, or could they eventually learn to suppress this behavior, which gave them nothing, no food, and reach instead from the side, right? So a simple, flexible behavior. And what you get here in their original data is that the bigger the cranial capacity, so the bigger the brain of a species, and you have primates in red here and birds in black, The bigger the brain, the better the species perform. But look at this, you have corvids performing at ceiling level already, despite their tiny little brains. What gives? How do you explain that? Well, if you take the same data and plot them now, plot performance as a function of the number of cortical neurons, regardless of the size of the brain, regardless of the size of the species, you get something that looks much better, and that where birds are actually now overlapping with primates, and corvids perform at ceiling level together with monkeys and other great apes. It's like the more neurons you have, the more flexible your behavior can really be, which is really interesting, and of course got me into really hot waters when I published another paper saying that dogs have twice the number of cortical neurons than cats have. I got a bunch of emails saying, you're saying that it's okay to love dogs more than cats. I'm like, no, I didn't say that. But if we're going there, let's love macaws even more, because macaws have tons of neurons in their cortex, and maybe we could have a bit more sympathy for raccoons as well, because think a dog that can learn all the tricks that you want to teach them, and they have those crafty little fingers to open all sorts of devices. But anyway, so that's the idea, that the more neurons in the cortex that a species has, the more cognitive flexibility it has, which I like to propose that is a good practical working definition of intelligence, as simple as that. It's cognitive flexibility. Can you change your behavior when circumstances change? It's not simply adaptation. Adaptation is passive and retrospective. Flexibility, cognitive flexibility, includes being able to act towards your future. So great, more neurons, more cognitive capabilities, makes sense so far. What about humans? Is our brain any special in its number of neurons? The answer is a big resounding no, at least so far in every single way that we've looked. The thing is, you have to compare humans to other primates, which is what we are. And then you find that the volume of the gray matter of the cortex of the human species is exactly what you would expect, what you would predict for a generic primate that had the number of neurons that we have in our cerebral cortex. It's not a fair comparison at all to non-primates because their brain is made in a different way. I'll get to that in a second. If you look at the volume of the white matter, so where all the connections are, which is something that people have said, that is what makes us humans special. We have much more connectivity in the white matter. No, we don't. It's we have the volume of white matter that you would expect in the primate, in the cortex of a generic primate with our number of neurons again. Within that cortex, the number of prefrontal neurons, so the truly associative neurons, neither sensory nor effector, it is a constant fraction of the total number of neurons in the cortex within primates. And again, this is the line plotted without humans, and you see where we fall in terms of how many neurons you would expect to find in the prefrontal portions of a generic primate that had this many neurons in all the other parts of the cortex were that generic primate. And again, the volume of the white matter, of the associative white matter underneath that prefrontal cortex, once more, exactly what you would expect for a generic primate that had as many neurons in the cortex as we do, which is 8% of neurons in the cortex as a whole. So those are, just to give you an idea, those are the numbers that we find in some primate species that we counted directly. So I just showed you that birds, a particular type of bird, crows, they perform in cognitive tasks that require flexibility. They perform as well as great apes already, even though they have a much smaller cortex. How do you get that? Well, birds have a prefrontal cortex too, even though it cannot be called a prefrontal cortex because it's not in the front part of the brain, much less more anterior to it. It's in the back of the brain. But if you look at the functional connectivity, it's also a highly associative portion of their cortex, even though it's not obviously layered. It actually is layered if you use the right techniques, but another whole different story. So the question is, there is this part of the bird pallium called the NCL, needlecaldum laterale. The name is in Latin and I'm butchering it, but there's this part of the bird cortex that is entirely associative. So what do we find? How many neurons do we find that there in comparison to mammals? And let me say this again, when we know that the overall connectivity of the brain is equivalent across birds and non-birds. So what we find is that a much larger proportion of all neurons in the cortex are actually associative neurons in the bird brain. And the absolute numbers actually place corvids on par with chimpanzees and monkeys. They're right there in between chimpanzees and monkeys. So that's a whole lot of neurons. How do we know that there are this many neurons in different species? That's really important for the story that I'm going to tell you next. So I just want to make sure that you know where these numbers come from, because the numbers that I've been talking about, these numbers here, these are actual numbers. They're not just ballpark estimates or educated guesses. These are actual counts estimated directly from the brains of different species. So how do we get the data that I'm going to show you on numbers of neurons in different species? We literally turn their brains into soup. You can use the brain as a whole, but you can also dissect that brain into the parts of interest. So from here on, I'm going to be talking about numbers of neurons in the cortex as a whole, and then numbers of neurons in the hippocampus. That means the entire dissected hippocampal formation. So not just CA1, CA3, but the whole formation. But anyway, so you take your structure of interest, or you take the whole brain and break it down into structures of interest. You chop those structures. Yes, it really looks like a kitchen recipe. And then you mush that structure. You mush up that tissue in a detergent solution that dissolves the whole tissue, the whole cell, all of the cell membranes, and leaves you with a suspension of nuclei that looks like this. And so it looks like cloudy, unfiltered apple juice, which my students tell me I've ruined for them. But the important thing is that in this volume of cloudy apple juice looking brain soup, you have all the nuclei of all the cells that were in the structure that you started out with. And they're floating in a volume of your choosing, which means that the estimate that you're going to get next is completely independent from the size of the original structure of interest. And that's really important, because if you want to use those numbers to study diversity and to study scaling, meaning to study how do they change with the size of the structure, you need to have an independent estimate. And that estimate comes from then taking aliquots of that suspension to the microscope, stained with, you stain the suspension with DAPI, take them to a fluorescence microscope that can also do white light at the same time. We just had an interesting problem in Sydney trying to get this to work, because the first microscope that we had access to was too modern. So we couldn't turn on the fluorescence and the white light at the same time, because who in their right mind would want to do that, right? But if you can do that, then you see the grids and the fluorescent neurons at the same time, which allows you to very, very easily, in a matter of 10 minutes, you have the counts. And This is what they look like. So you see that it's very clear what is a nucleus, which means that, and this is the key assumption that we're making, it means that as long as every cell in our tissue has one and only one nucleus, what we're doing when we're counting nuclei is we're actually counting cells. So the DAPI gives us the total number of nuclei in the tissue. And then we do a second step with antibodies, in this case, NUN, that for our practical purposes, we can consider to be a universal and specific marker of neurons. Let me go back and forth, and you can see that it's very easy to tell what nuclei are positive for nuns, and therefore were neurons, and everything else is not a neuron. What they are, some sort of glial cell or vasculature, whole other story there again. So with these numbers, we could, I've been fortunate to be able to find people in different parts of the world who studied different species and who were willing to collect some of their brains and give them to me to turn them into soup, which yes, took some convincing in some cases, especially when you approach like, hey, I learned you have 4 elephant brains in your lab. Could I have one, please? Just a hemisphere will do, but I want to turn it into soup. So you can imagine how the conversation goes from there. But this is really important because you need the diversity to be able to say what is really going on, how the different brains really compare. Note that you cannot just, I mean, you can, of course, you always can take a mouse brain and a human brain and say, this one has 1,000 times the number of neurons that this one has. Yay. So what? Does that mean that the human brain is special compared to the mouse? Does it mean that it's just a primate with a big brain? Does it mean something else? You need the full picture. So let me give you a flavor of what you get when you look at the full picture, because that's what spurred the whole rest of the story that I want to tell you about longevity that eventually takes us to the hippocampus. So when you compare, come on. When you compare different species and you line them up by numbers of neurons in the cortex, what you see here is that very few species have, my thing seems to be frozen. You see that very few species have large numbers of neurons well above, There you go. Start again. Okay. So you see that very few species have more than 1 billion neurons in their cortex, right? So either species with more than 1 billion, that's why it's stuck in the next one. I don't want the next one yet. Oh, technology. There, species that have mammalian species with more than 1 billion neurons in the cerebral cortex, they're either very large or else they're primates, which gives you an idea already that their brains, their cortices are made according to different rules. And the different rule has to do with what happens to the average size of the neuron as you get more neurons in different species. So the result of that is that if you rank species up by body size, which is what people usually do when they look at diversity and ask themselves, is there any order behind this diversity, right? The size of the body is the low-hanging fruit, so you go for that. So when you rank species, mammalians, well, actually I have birds here too, that's how you find, that's what you get in the ranking, and you see humans placed nowhere really catchy or that we would be really proud of, right? But change the ranking now for numbers of neurons. And what you're going to see, if this thing collaborates, come on, is that, there you go, that humans actually have the largest number of neurons in the cortex. Again, not because we're any special compared to other primates, but simply because amongst the primates, we have a cortex that's made like the cortex of all other primates. It just happens to have more neurons than all of them. And let me give you an idea of the numbers here. So in a giraffe cortex, you find 1.7 billion neurons, which is about as many as you find in the cortex of a macaque monkey, a rhesus monkey. 1.6, even though the giraffe cortex is much bigger than the monkey cortex. And actually you find even more neurons in the cortex of a macaw. And when you come to humans, you find that even though our cortex is only half the size of the cortex of an elephant, it actually has about 3 times the number of neurons. So 16 billion neurons in the cortex compared to the elephant. And like I said, the difference in how many neurons you find in the cortex of different species comes from what happens to the size of the neurons in different species as they gain more neurons. In non-primates, typically the more neurons you find in the cortex, the bigger those neurons are. Not true in primates, and even less true in songbirds and parrots, where the average size of the neuron is really, really small, much smaller than in primates, which means, of course, that if you compare brains of similar sizes, cortices of similar size, you're going to find that the bird has many more neurons in the cortex than a primate, which has many more neurons in the cortex than any other kind of mammal. Good. So this is really key because when you look at the raw data, this is what it looks like. And it's the shape that led to the whole rest of the story that I'm telling you today. So here you're looking at, and this is what I'll be showing from now on. Every dot is one species. When I have birds, they're going to be in black. The colored dots are the mammals. Primates are always in red. And so you can tell the human data point apart from the other ones. It's simply the red point with the highest value. So here you see that larger animals tend to have larger brains, sort of. There's some variation here, but there's definitely a consistent trend. The bigger a mammal is, the bigger its brain tends to be. So you would expect, if all else were made equal, that inside that bigger brain should be a bigger cortex, and inside that bigger cortex should be more cortical neurons. So the bigger an animal is, the more cortical neurons it should have, except that that particular graph actually looks like this. So now you're looking at the number of neurons in the cortex and how it varies with the size of the species, the size of the body of the species. And what you see here is that, well, do bigger species tend to have more neurons in the cortex? Yes and no. Yes, definitely yes. If you're comparing species that can be compared to each other, that species that belong to the same clade. But no, if you're comparing, say, a primate to another type of animal, or a songbird or a parrot to even a primate. So for a similar body size, what we find is that birds consistently have more cortical neurons than primates, which consistently have more cortical neurons than other non-primate types of mammals, right? So that's a really important pattern because that was the pattern that you're going to see when we get to longevity. But first, let me tell you how we get to longevity, or how I got to from this to asking questions about longevity. Remember, I told you the third question I'm interested in is, so what that you have diversity? We usually think in terms of having more neurons as what it gives you for cognition, right? But You can also think of neurons, and I like to do this when I talk to non-scientists. I like to think of neurons as the Lego pieces that build brains. They're the functional blocks that build brains. And what you see here is that when you have just a handful of Legos, or you have a small bucket of Legos, or when you have a truckload of Legos, you can build completely different things. The more pieces you have, the more flexibility you have in what you build, the more complexity you can build, but also the more time it's going to take you to build that structure. So, and I fully acknowledge that this was a question out of nothing more than simple intuition and looking at our real world and saying, well, whenever there are more pieces to go into a whole, into a system, putting that system together takes more time, right? And whenever I talk to people who work in, who are computationally minded, I ask them if this makes sense, and they reassure me that yes, this makes perfect sense. The more units you have in a network, if you're gonna let that network train itself, which is what our brain does, what's what brains do, the more units in that network, the longer it's gonna take to train that network and reach a set functional state. So the question then, do we have, does it take longer to build a brain that has more neurons? Well, how do you address that? Long story short, I realized that the time it takes for an animal to reach sexual maturity should be, at the very least, should be an indication that by that age, that animal is functional enough, it's cognitively independent and capable enough that it can produce offspring, right? At the very least, it can produce offspring and not kill it, which is a really important thing in terms of evolution. So that is called life history. And that is a really, really important topic, because life history, the other side of life history, is longevity. How long do animals live? And conventional wisdom has it that that is determined by how much energy your body uses, and that's your metabolic rate, and that is related for instance, the frequency at which your heart beats, which makes for this idea that everybody, doesn't matter how large or small, has about 1 to 2 billion heartbeats in them, which is a really scary thought, right? Every second, you're one heartbeat closer to your final demise. But the overall story is this here, that a slow burning flame must burn longer. Which means that the more slowly your body uses energy, the longer it should take all the biological processes in your body to happen, including reaching sexual maturity and then living your full lifespan. Now, the problem is, I think, in modern days, we don't live with farm animals anymore, because if you do, you know that cows and horses are much larger than we are, so they should take much longer to mature. And yet they're mature in just a year or so. In my case, I don't live with a proper horse, but I do live with this little guy here that while it took me 16 years to reach the size that I have, he did that in just 9 months. We weigh the same. So that's at the very least an indication right there that something is not right. Typically, the way people respond to that is, well, yeah, humans are different. Humans are special. Humans take much longer to develop, and humans live much longer lives, right? So let's see what we find when we actually look at the data. And let me walk you quickly through how we got there. So I found the database that I needed, which was a database of life history in different species, whole lots of different species, thousands of them, literally, that I could then add my own data on brain size and numbers of neurons in the different species and ask the question, does it take longer to mature a brain that gets more and more neurons in the cortex? So let me just give you a sense of where things were. So this was the conventional narrative. The longer it takes an animal to mature, the longer it also lives somehow. And this somehow is thought to involve selection. You're selected for, it's a long argument. I won't walk you through it, but let's just leave it at that. Whenever you're selected for a longer life, you're also selected for a slower sexual maturation. The important part here is where humans are, which is not very far at all from the expectation. But so let's break this down and look at the idea for metabolic rate and how metabolic rate should have something to do with how animals age and develop, how quickly they age and develop, and how long they live. So here you're looking at each point is again one species. We're not coding them for anything, just vertebrate species. So here you see that the lower the specific metabolic rate, so watts per gram of tissue, of body tissue, the lower the specific metabolic rate, the longer an animal lives. And let me be clear, when I say longer, the longer they live, I'm talking about maximal longevity, not average longevity. Each of them has its shortcomings, but maximal longevity, for a number of reasons that I can tell you later, I think is the less problematic variable here. But anyway, so the more slowly a species uses energy, the longer it seems to live. And let me point your attention to this value here, the R square. This is definitely a significant correlation with an R square of 0.23, which means that you can explain 23% of the variation in longevity across vertebrate species, across warm-blooded vertebrate species, through the variation in their specific metabolic rate, right? Slower churners live longer. Great. Now, a really handy thing is that we know that for reasons that are not quite clear to this very day, if you look at warm-blooded species, these are the cold-blooded species, the bigger an animal is, the more slowly it uses energy, with a much better correlation coefficient here that explains 40. So body size explains 46% of the variation in metabolic rate. Why do we care about this? Because measuring metabolic rate is really hard, but measuring body mass is trivial, which means that you can look at those two things directly, and because they have the specific metabolic rate in common, you can then test your hypothesis through the correlation. By seeing if the necessary correlation is there, you can test your hypothesis that the bigger an animal is, the more slowly it uses energy per gram of tissue, and therefore, the more slowly it accumulates damages, and therefore, the longer it lives, right? Up to that point where it just finally can't anymore. And that gives you this plot here, which is a very significant and pretty decent variation of longevity with increasing body mass, where body mass variation accounts for 32% of the variation in longevity. Remember that this is biology, and 32 sounds like a very decent correlation, right? Except that what happens if you're somebody obsessed with patterns, and especially the patterns in diversity, some of which I've shown you already. If you color code the data, this is what you find, that, well, yeah, the bigger an animal is, the longer it lives, but for a similar body mass, birds live longer than primates, who live longer than non-primate mammals. Sound familiar? The instant I saw this graph, I went, get out of my way. I cannot do anything else. I have to do this. And I will not be separated from my laptop until I'm done with the data analysis. So this is where humans are, a point outside the curve. So this seems to support the idea that humans are extraordinarily long-lived. Except that if you compare humans to other primates, things are not quite the same. So here's that same graph that I showed you beforehand. And let me walk you through the main findings here, because the question then is of course, if longevity is related in clade-specific ways to body mass, and numbers of neurons in the cortex are also related in the same clade-specific ways to body mass, then maybe body mass is just irrelevant. And what I'm looking here really is a correlation between numbers of neurons in the cortex and longevity, right? So I did that with the actual data that the 74 species that we had. And that's what you get. You get a much better correlation, even though it's fewer data points, where you explain 74% of the variation in longevity through the variation in numbers of cortical neurons. This is what the same data look like if you look at longevity as a function of body mass. So the explanation goes down to 23%. And if you use other parts of the brain, like cerebellar neurons, you find that birds and mammals do different things. If you use the rest of the brain, which are all those parts of the brain stem all the way up to the hindbrain that really, truly connect to the body, which was my original expectation, right? This is where it should matter, how many neurons you have. The more neurons you have here, the more resilient your system should be to any damages incurred. But you see that there's a very clear, clearly different pattern between birds and the other, all the other animals. And here again, you see what you find for cortical neurons, which is much better relationship. Now, that 74 species doesn't compare really to the couple thousand species that the whole data set had for body size. So you could say, well, that's not really fair, is it? You have so many more data points for mass. You should do the same for neurons. Short of going out there and collecting hundreds and thousands of different species, which would be great, but not feasible, at least not right now, the next best thing that we can do is use the math that we learned that describes the relationship between, and describes very, very well, typically with R squares of 90 something, the relationship between the size of the brain of a species of a certain clade and how many neurons that species has. So we can use these predictive relationships and apply them to the size of the brain of different species, and therefore estimate how many neurons they should have in the cortex, right? And then I can apply those equations to transform brain mass in this plot here, which only explains 48% of the variation in longevity, and get numbers of neurons predicted from that. And if numbers of neurons in the cortex have nothing to do with longevity, then all that I'm doing is introducing noise here, right? And the result should be like a buckshot graph with data points all over the place. Unless if there is a true correlation here, and in which case you would expect bringing those different equations, clade-specific equations, to make the whole thing collapse, which is exactly what happens. So when you estimate data, estimate brain size, numbers of brain neurons in, this is 705 species, you actually maintain the same level of explanation of it that you can do for variation. Now, the really interesting thing is that we can do residual analysis and use the residual of the longevity. So what this means is we can calculate how much longevity for each species is not explained by longevity in body mass. So that's the distance between each point and the curve here. And we can see if there's anything left to be accounted for, any residual longevity unexplained by body mass that can be explained by variation in the number of neurons in the cortex. And the answer is, yeah, there's still 58% of the variation that goes unexplained by body mass. Now, so let's do it the other way around. If we calculate the residual, so what is left to be explained by after you've accounted for numbers of neurons, is there any residual longevity? Is there anything left to be accounted for, to be explained by variation in the size of the body? No, the size of the body is essentially irrelevant. No power, almost literally zero power. Same thing with specific metabolic rate. Once you account for numbers of neurons, the specific metabolic rate, you find there's no signal left in the specific metabolic rate to explain that variation. So what I have shown you so far is, and of course you can do the same thing for sexual maturity. What I've shown you so far is that the more neurons a species has a warm-blooded species has in the cortex, the longer it takes to reach sexual maturity, and the longer it lives after that, and humans are no exception. Now, what I can add to that, and that starts with, let me draw your attention to this bottom graph here. So this is a paper that's been a long time in preparation. It's the older people in the room will certainly be able to relate to this. You start a study and then things happen and then you just never get to actually sitting down and finishing writing the paper. So these data have been around for a while, and which show that there is some clade specificity in the relationship between the size of the hippocampus and the size of the rest of the cortex. Another really important part here is that the cortex gains mass faster than the hippocampus, or so if you turn it around, the hippocampus gains mass more slowly than the rest of the cortex, which means that in evolution, as you get animals with bigger and bigger brains, it's really the cortex that's ballooning on top of the hippocampus, right? Except that if you look at numbers of neurons, you find that there is a set relationship between numbers of neurons in the hippocampus and the number of neurons in the other parts of the cortex, right? Non-hippocampal cortex. And really important to me, you notice the exponent here. Okay, so what I forgot to draw your attention to is that all of these graphs that I showed you, they're these beautiful scaling relationships. They're not linear. They have exponents of around 0.6, 0.7. Why do I care so much about a relationship being linear? Because a true cause of change in Y has to be linearly proportional to Y. We can talk about that later, but that's just simple math right there. If there's a causal relationship, there needs to be a linear relationship, which means that cortex as a whole, if it's not linear, then it's not the cortex as a whole. So our candidate of whatever it is that could really determine how long it takes for a cortex to assemble and how long that animal around the cortex lives has to be a linear function. And now that we know the relationship between numbers of neurons in the hippocampus and numbers of neurons in the cortex, we can do the same kind of transformation that I just showed you. We can estimate numbers of neurons in the hippocampus, and we can test whether we have a decent linear predictor of longevity. So let me start with this one here on top. So these on the left, you see the scaling relationship. So we're plotting power functions, and you get power functions for both cortex and the hippocampus, with a larger exponent that's closer to 1 for the hippocampus. Now, if your question is, well, yeah, sure, but is this power relationship also reasonably linear? Which it can be mathematically, that's a possibility if this exponent can actually also be one. If you try to fit a linear relationship to the scaling of longevity with numbers of neurons in the cortex, you get something that you could force your way to say that that is a decent linear relationship with an R square of 0.54. If you use hippocampus instead, the number of neurons in the hippocampus, what you get is something much more decent that applies very well to almost the entire range, just not so much to the smallest mammals in the data set. And I think there's a good possible explanation for that, which is temperature. But anyway, let me just finish walking you through this story. Notice the R square of 0.78. So 78% of the variation in longevity can be accounted for by variation in the number of neurons in the hippocampus. Even more interesting is that we can work out the math. The math of that linear equation boils down essentially to every million neurons added to the hippocampus of a species means, or it comes with, not saying causes, but it comes with 40 extra days to reach sexual maturity and 1 added year of life. Let me say this again. Amongst warm-blooded species, and I'm talking mammals at this point, the more, so there's variation in how many neurons you find in the cortex, there's variation in how many neurons you find in the hippocampus that may or may not have something to do with the size of the body. If you go broad, there won't really be a universal relationship, but the more, what difference does it make that different species have different numbers of neurons? The more neurons you have in the hippocampus, the longer you live. And the longer it takes you to get to that point where you can reproduce. There are so many interesting consequences of this. My mind, of course, goes instantly to human evolution, because that means that if the number of neurons in our cortex tripled over the last 2 million years, that means that not only we gained in cognitive capacity, but we also gained in the length of childhood, which means that we have more and more time available to learn by imitating others, by imitating other kids, by interacting with other kids, by interacting with our elders. It also means that at the same time, our elders live longer lives and they can be around us for much longer, right? So there you have this interaction between capability that brain capability that depends on your numbers of neurons, I like to think, but that through time, as you have more time, you gain more and more possibilities of learning to use those neurons, learning to develop that into something like a culture. So now the question, of course, is how could that possibly happen? So let me leave you with my working hypothesis here, which, if anything, it's testable at least. And essentially the hypothesis is that damages accumulate over time, no matter what, actually only depending on the temperature of your body. And all warm-blooded animals, the same rules apply to all warm-blooded animals that really function at about 37, 38 Celsius. So if that is true, that damage actually accumulates over time at a similar rate, and there's evidence that now from independent groups that the rate of aging really doesn't scale. It's very much the same across warm-blooded individuals. Then there's an advantage in a way, if you think that a longer life is a good thing, there's an advantage in starting your life with more neurons in the part of the brain that does that really important integrative job of keeping your whole together, which means that if fractional damage, if the rate of damage is the same, as time goes by, the whatever species starts out life with a very small number of neurons in the hippocampus will very quickly reach a point where there's hardly any left that are healthy enough to keep the body integrated. So that's the two yellow neurons that are still yellow in the small guy over there. But in the same amount of time, if you started out with a much larger pool of neurons, you've gained resilience. Your system is still capable. It's still functional. It can withstand damages for longer and longer intervals of time until it eventually gets to that point where it breaks down. So that's the testable hypothesis here, that more hippocampal neurons give the owner of that brain more resilience to withstand the damages that inevitably occur as we live in a similar temperature, with a similar body temperature, and in which case we can really think that it's not that humans are uniquely prone to age-related, or I would say time-related diseases, but rather that humans are fortunate enough to live long enough that we get to the point where our brains accumulate so much damage that some effects start appearing in the brain that you wouldn't see otherwise. So I'm going to finish here and thank my collaborators who made working with figuring out the numbers of neurons in all these species possible. And thank you so much for your attention. And I hope there's still time for some questions. Thank you.
So where do ectotherms sit in this model? I mean, they might not have thermoregulation like we do, but they probably end up both hotter and colder and have accumulate potentially at least as much, if not more damage, but they're very long-lived, some of them.
They're right here. So for a similar number of neurons, they live longer lives. We just got the data, which is exactly what you would predict in a simple Arrhenius chemistry world, right? Where the rate at which things happen, the rate at which damage happens simply depends on the temperature of the system. Doesn't matter how quickly or how slowly you actually use the energy, it's the temperature at which life happens.
Thank you. An interesting hypothesis. If you take a non-comparative perspective and look at humans with hippocampal atrophy, what are your perspectives about longevity in humans with hippocampal atrophy?
Well, you would expect that they would have shorter lives.
And
Do you know
evidence?
the answer? I don't know the answer.
I don't think there's a linear correlation, but it's an interesting thing to get the evidence and look at this hypothesis from a completely different perspective.
Right. So the prediction there, I mean, there's manifold, right? Well, it starts with, what I described to you, does it also apply within the species for individual variation? And the complication there, of course, is that there are all these other things that you can do with your life once you're born with a certain number of neurons, right? So I would expect that itself will bring a whole lot of variation that you cannot expect to be accounted for by the number of neurons in the hippocampus. But still, this is a very easily testable hypothesis, because there are different strains of mice that have different ages. So the prediction that they have different longevity. So the prediction there is that there should be clear differences and correlations with the number of neurons in the hippocampus of those species. And yeah. So, but I think what I'd like to point out is that at least now we have a working hypothesis, a testable hypothesis for how come things that we can do, like cutting calories and exercising, that we know that they benefit the health of the hippocampus. We also know that they prolong healthy lifespan. Does it do that because they improve hippocampal life? If that is true, if there really is a correlation there, then we've just learned that we can really choose to act, to do things with our own lives that improve hippocampal health, and we will be acting towards our own healthy lifespans.
That's a really fun concept, and it makes me think of two things. Number one, London taxi drivers, not Uber drivers, who had expanded hippocampal volumes. That doesn't necessarily mean neurons, but I assume it does mean so. That's one. And number two, I saw a paper recently, which I think is quite fun. If you take gamers, habitual gamers, and you give them a spatial memory task, and you compare that to people who are not gamers, the non-gamers use the hippocampus to solve the task, and the gamers use the striatum. Are we dooming our species with gamers?
I think those are really fun questions to ask. They're really important questions to ask. I'm reminded by my social scientist daughter that answering them is absolutely not trivial. Fun as it would be, but I think it's great that we have clear predictions to make, right? And difficult experiments start out with anecdotes and interesting stories that catch our attention. So maybe somebody here will be inspired to, or knows a social scientist and who can talk them into running properly one of these difficult studies. Definitely would be fun. So for now, what we can do is use what we can control and which is mice, right? Always the mice, which is, I would add, a wonderful reason why it's great to find out that humans are not special, right? Because that means that anything that has to do with numbers of neurons, anything that depends quite simply on numbers of neurons, if it really does depend simply on numbers of neurons, mice should be a great model, except if you're looking at, if you're studying diseases like Alzheimer's that take 60 years to develop, that take the accumulation of damage over and over and over, decades of life. Can you really expect that in 2 years, a mouse is going to accumulate as much damage as a human cortex can in not even 90 years, make it 40 years? I don't think that's reasonable. But then there's tricks that you can use if once you understand what's going on. If it's true that it's the time-related accumulation of damage that is a factor, then now there are mice available that have enormously increased rates of damage accumulation of DNA damage and other types of damage in their cells. They live much shorter lives. They look like no regularly old mouse will ever look. They look terrible. Maybe that is a much better model for age-related diseases.
So just as another example species, so dogs, which vary wildly in size and in lifespan, how do they track with size of the hippocampus?
That's a really interesting example of intraspecific variation, because dogs up to a certain point, they live fairly long lives, about what you would expect, around 15 years, absent diseases, right? Except for, sadly for me, Great Danes, which seem to overall have much shorter lives, maybe best case scenario, 11 or 12 years. But there's a very well-documented reason for that, which is gigantism itself, which is related to insulin receptors and insulin desensitization and all the things that get screwed up in the body with that. So, but it's interesting because it's one of those cases that at first look like a contradiction, right? Or a negative result. And maybe they're actually the kind of exception that proves the rule. Because yes, overall, generally, this is what happens, unless you have a condition that damages, that does much higher rates of damage to your relevant neurons. Let's say that they are the neurons in your hippocampus, right? But yeah, definitely something that needs to be addressed.
Thank you very much.
Thank you so much for your attention.